Hello students, welcome to EPG Pathshala. I am Dr. Hari Prasad, faculty at the Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today we are going to talk on the module Principles of Protein Structure Prediction under the paper Bioinformatics. After completing this particular module, you should be in a position to understand genes and proteins, protein structural organization, protein folding, Anfinsen's experiments and lessons, Levinthal's paradox, energy minima and stability of protein folds, methods of determining protein structures, X-ray crystallography, the essentials of NMR, Cryo-EM, small angle X-ray and neutron scattering, their principles, advantages and disadvantages, protein structure prediction, the principles and advantages, and the uses and applications of protein structure prediction. When one talks about protein structure prediction, it is important to be familiar with two molecules. They are the genes and the proteins. The genes and the proteins come together at a particular interface, what you call as the central dogma. What is the central dogma? DNA executes its function through complex macromolecules called as proteins. The basic fundamental unit of protein is amino acids which are linked up together by peptide bonds. There are a total of 20 amino acids that could be constituting a particular protein. The journey from the gene or the DNA to the protein is a complex and a tightly controlled mechanism within each cell. It consists of two major steps, transcription and the translation. Together, these two fundamental roles of transcription and translation are known as the gene expression. During the process of transcription, the information that is stored on the DNA is transferred to the messenger RNA that then carries the information from the DNA out of the nucleus and into the cytoplasm. Once in the cytoplasm, the messenger RNA interacts with a specialized complex called as the ribosome, which reads the sequence of messenger RNA bases called as codons that codes for a particular amino acid. A type of RNA called as the transfer RNA assembles the protein by putting together one amino acid at a time until the ribosome encounters a stop codon. The flow of information from DNA to RNA and finally to the protein, which is now a well-folded three-dimensional structure with a particular function, is a very fundamental principle of molecular biology and is called as the central dogma. Structural organization of proteins. Proteins are made up of numerous small units called as amino acids which are bound to each other in long chains. Amino acids are biologically important and contain amino and a carboxyl functional group and the most distinguishing third group called as the side chain. Different side chains give amino acids different properties. The carboxyl group, the amine group and the side chain and a single hydrogen atom are all bound to a central carbon atom called as the alpha carbon. Broadly, 4 different levels of structural organization is found in proteins. They are the primary, secondary, tertiary and the quaternary structure. The primary structure comprises of a sequence of amino acids linked to each other by a covalent bond called as the peptide bond. Sequences which are less than 50 amino acids are generally referred to as peptides, while proteins that have more than 50 amino acids are called as proteins or polypeptides. Next, the secondary structure. Secondary structure is a three-dimensional arrangement of amino acid chain that is maintained by hydrogen bonded connections made entirely between the NH group of one peptide bond and the CO group of another peptide bond. It defines the conformational presence of a local region of a polypeptide or a protein. The structures that are considered or most seen in secondary structures are the alpha helix and the beta pleated sheet. Tertiary structure. The tertiary structure is the final specific geometric shape that the protein assumes and is determined by a variety of bonding interactions between the side chains of the amino acids. These interactions, which comprise of van der Waals interactions, hydrophobic interactions, ionic interactions, and also the covalent disulfide bonds, help in packaging the secondary structural elements into a very compact globular three-dimensional structure. And finally, protein quaternary structure is the number and arrangement of multiple folded protein units in a multi-subunit complex. It includes organizations from simple dimers to large homo-oligomeric structures like in collagen, or a heterooligomeric or a polymeric protein where you have two alpha and two helices coming together like in hemoglobin. These subunits are held together by variety of interactions to help in stability. Protein folding. When we talk about protein structures, it becomes important and pertinent to understand the concept of protein folding. Protein folding is the physical process by which a polypeptide, which is otherwise a linear chain of amino acids, folds into a very characteristic three-dimensional structure that has a biological relevance. The figure to the left indicates the polypeptide chain, and the figure to the right shows the well-defined three-dimensional structure. This particular three-dimensional structure is stabilized by a number of interactions that take place between the amino acids. The interactions could be between each other, or it could be the interactions of these amino acids with the water molecules. These interactions, whether they are van der Waals, hydrophobic, or ionic interactions, help to stabilize the three-dimensional structure of the protein, and this particular folded protein is called as the native state. Having understood protein folding, we then go on to understand what were the very first experiments that established protein folding. And the first one that comes to our mind is the Anfinsen's experiment of the protein folding using a ribonuclease enzyme. In the year 1973, Christian B. Anfinsen and his colleagues performed the definitive experiments showing that protein takes its specific shape based on the directions encountered in the sequence of amino acids. So, in the first experiment, he took a ribonuclease enzyme, which is a well-folded protein with 4 disulfide bonds connecting 8 cysteines. This particular protein was reduced using beta-mercaptoethanol and denatured using 8 molar urea. The harsh conditions of beta-mercaptoethanol and 8 molar urea denatured this particular protein. Subsequently, when this denatured protein was subjected to dialysis to remove out the 8 molar urea and subsequently oxidized, we find that the protein refolded and almost close to 90% of the molecules were in the active form. In the control experiment, the same set of experimental conditions were conducted up to the denatured state. After the denatured state, for the refolding, oxidation was done first, followed by removal of 8 molar urea by dialysis. What do we find now? We now find that almost all the molecules are in a scrambled state and just 2% of it is active. What is it that has happened? In the experiment, what needs to be understood is the fact that when refolding was taking place with the removal of 8 molar urea, the protein folded to its native conformation and the oxidation helped or facilitated the formation of disulfide bonds between the cysteines which came close to one another. This resulted in an active protein. While in the second case, when the denatured protein was tried to be refolded, the oxidation meant that the disulfide bonds were already formed between certain cysteines which came close to one another, and it really did not matter that 8 molar urea was subsequently removed because it continued to stay in a scrambled state. And the 2% active molecules are the result of a probable chance where the right cysteines came close to one another to form the right disulfide bonds. This actually demonstrates the fact that fully active enzyme and the transition is driven entirely by the reduction in free energy that occurs in going from a scrambled to a catalytic form of an enzyme. This demonstration that the shape that a protein realizes in solution is dictated by amino acid information is expressed in terms of thermodynamic stability. Lessons from Anfinsen's experiments. Having understood Anfinsen's experiments, the take-home messages would be: 1. The information for the folding of a protein is within the amino acid sequence. 2. It is possible to fold a protein in vitro given the right conditions. And 3. Disulfide bond formation occurs between free cysteines that come close to one another. Levinthal's paradox. As we have now understood, proteins have an incredible number of possible conformational states, but they are still able to fold very quickly. When I say very quickly, I am telling in terms of a fraction of a second. So as you see, the figure on the left is a very random coil arrangement which takes up a very beautiful three-dimensional structure that is depicted on the right. The protein folding process is speeded and guided by the rapid formation of local interactions which determines further folding. Also, this suggests local amino acid sequences which form stable interactions serve as nucleation points in the folding process to result in a native state. What do we have here? We have a cute little boy who is playing with a toy panda. What is he doing? He has just pushed gently the particular toy, and what is it that we see? We see that this particular toy is moving to and forth and looking for various possible positions at which it will remain stable. This particular toy panda is special for the fact that it has a very spherical base. Therefore, it continues to oscillate, it continues to move slowly but steadily to reach that particular stage at which it is most stable. That the gravitational point is so placed that this particular doll would be stable only in one particular position. You see, now the toy is almost coming to a standstill position. That would be the most stable position. You see, it has now almost reached that particular stable position. Now let's see what the boy does. He has moved it and is giving a bigger push to the particular toy. Yes, even irrespective of whether it's a gentle or a forceful push, the toy continues to oscillate and will until that point of time where it reaches a stable position. And once it has gained that particular position, will continue to stay that way till the end of time. This particular aspect of the doll trying to reach a particular stable position becomes very important in understanding the concept and the phenomena of protein folding, which we will subsequently study or understand. So having understood about the aspect of the doll that was trying to look for a stable position, we now come to the concept or understanding of protein folding. What is it that we see here on the slide? We see that a chain of polypeptide with a number of amino acids is trying to look for a particular stable conformation. It continues to fluctuate between different positions, and as it has not found a stable position, continues and tries a series of different conformational positions. As you see, the secondary structural elements are being formed. Once these secondary structural elements of helices and beta pleated sheets are formed, they are trying to see what packing or the aspect they are supposed to interact with that will give it a very compact, minimal energy, stable state. Just as in the case of a doll, it will continue to do so until it reaches the most stable state. And this particular state is called as the native state of the protein. In continuing to understand protein folding, we now move on to understanding protein folding from the perspective of energy landscape theory. According to this, protein's native state corresponds to its free energy minima under certain solution conditions. As you see, it's a broad-based funnel which has the random coil conformations, and as the energy falls, you get into the more narrow and the deeper aspects of that particular funnel. Partially folded proteins become trapped, creating energy landscapes due to non-native local minima in the middle of the funnel. The deepest end of the funnel has free energy minima corresponding to single, well-defined tertiary structures, which is represented by a red protein molecule. The driving force for the protein folding is the stabilization associated with sequestration of hydrophobic residues in the interior of the protein, which allows water to maximize its entropy, thereby lowering the total free energy. The molten globule state, which is shown slightly above the native state, as predicted by the folding theory, is an ensemble of folding intermediates, thus corresponds to a protein in which the hydrophobic collapse has occurred, but many native contacts or close residue-residue interactions are represented but are still to form. Now, having understood what is protein structure prediction, we need to understand what are the possible experimental ways of determining protein structures. They are X-ray crystallography analysis, nuclear magnetic resonance spectroscopy, small angle X-ray and neutron scattering, cryo-electron microscopy, and our own computational based protein structure prediction. Now, Coming to X-ray crystallography analysis. X-ray crystallography is used to study the atomic and molecular structure of a protein crystal that causes a beam of incident X-ray to diffract into many specific directions. The measure of angles and the intensities of these diffracted beams provides information of the density of electrons within the crystal, thereby producing a three-dimensional picture of a particular protein. However, there are certain limitations to X-ray crystallography. They are that it requires a very large amount of pure protein. When I say large here, it's in terms of 10 milligram or more. You require protein crystals, and this becomes a very compulsory requisite. No crystals, no X-ray crystallography, no structures. Crystallization conditions are very time consuming and are very difficult and are dependent on a number of parameters, right from the concentration of the protein, the solvent, and including conditions such as temperature and gravity. Membrane proteins and hydrophobic proteins are very difficult to crystallize and are a challenge by themselves. Crystal structures are not classical representatives of in vivo protein dynamics. Crystals that diffract at low resolutions may not yield much information. And lastly, as I have already told, it's time consuming, laborious, and sometimes very costly. Nuclear magnetic resonance spectroscopy is another important methodology that is used in the determination of protein structure. What we see is peak positions that correspond to chemical shifts which mathematically derive the chemical environment of the nucleus. We see peak splitting that corresponds to coupling constant J, which is quantifiable as a peak separation and provides information about neighboring nuclei, or in other words, torsion angles. We also see peak intensity, which is representative of the integral function and is a ratio that gives the relative height of the integral curve and provides information of the nuclear count and T1 dependent. And finally, we also see the peak shape, which is representative of the line width and which biomathematical equation provides information on molecular motion and chemical exchange. Even though we have understood or seen how useful nuclear magnetic resonance is, it has certain limitations. What are they? It requires a fair amount of protein concentration. It shows very poor dispersion in HSQC spectra and the signals are very broad. Spectral line width is highly dependent on the molecule's tumbling rate in solution and therefore on its molecular weight. It requires large quantities of soluble protein. It requires multiply labeled protein. It is time consuming and costly and is best suited for understanding or studying small protein structures. Cryo electron microscopy and small angle X-ray and neutron scattering are few other technologies that have been used to understand protein structures. Let us just go through the principles, advantages and disadvantages of these two techniques. The principle of a cryo-EM is that individual particle 2D images of a molecule that is cooled is reconstructed to create a 3D structure using a software. The advantages of this particular technique are that the structures are hydrated native, it provides an Armstrong resolution, it provides structures of unstable complexes, and crystals are not an important requisite for this particular technique. On the other hand, that it has disadvantages that it is laborious sample preparation, the artifacts of the sample interfere with the technique, and the sample have to be electron transparent. Now, coming to SACS and SAN. The principle is that it uses elastic scattering at small scattering angles to investigate structures of substances. The advantage is that it has high penetration, reveals magnetic structure, and has resolution at mesoscopic scale of 1 to 100 nanometer. But the limitations are that it's available in reactors and is extremely costly. Advantages of protein modeling. When we talk about protein modeling, the primary or the important thing is the fact that the Human Genome Project is now completed, and that helps to provide a lot of raw protein sequence data. There is immense sophistication and algorithms that have come in place and is computationally driven science which is available to you at the click of your fingers. It's relatively more easy, less costly, and takes less turnaround time. Now, when you do experiments in the lab, it may be transcriptomics or genomics or whatever may be the case pertaining to the function of the protein, you can complement these wet lab experiments and assays with the protein structure analysis done computationally, thereby making science more efficient. Even though we use the word prediction, there is a scientific reasoning, there is a logic and accuracy that can be further validated and verified. Now, you have a protein data bank. It comprises of data or coordinates of more than 25,000 protein folds. Now, this particular information is very important and can be harnessed to good effect in our prediction models. Progress in this particular field will help to propel the future of pharmacotherapeutics, which helps to tell about potent drug designing and also the right drug for the right disease in the right individual. Now, if you ask me, what are the success stories in structure-based drug designing? I will say plenty. One, you have captopril. The target is the angiotensin converting enzyme and is used in hypertension. Then you have dorzolamide as a carbonic anhydrase used in glaucoma. Now you have nelfenavir, which has its target in HIV protease and is used in AIDS. Zanamivir acts against neuraminidase, an important protein in influenza. Sildenafil is a phosphodiesterase inhibitor and is used for erectile dysfunctions. Imatinib is a BCR-ABL kinase enzyme inhibitor which is implicated in the causation of chronic myeloid leukemia. In the same leukemia, you could have a mutation of this particular kinase where threonine gets converted at position 315 to isoleucine, and structural studies have helped to design dasatinib against this particular mutation in patients with CML having this particular mutation. And protein structure prediction has a number of uses and applications. To enumerate, structure function relationship of protein could be deciphered. You could do structural characterization of a drug target from various diseases. Structure-based drug designing, as I already explained, which includes potency and the right drug. You could establish the structural basis of a disease. You can understand the structural mechanism of drug toxicity. You could have a structural approach to overcoming drug adverse effects? Structural perspective to understand evolutionary background of protein is also possible using structure prediction models. And finally, this particular aspect of protein structure prediction lays a platform for personalized medicine, and this will only get better in the years and the decades to come. An important use of protein structure prediction is to understand structure-function relationship in proteins. I have provided an example of the structure-function relationship of a group 3 phospholipase A2 from the scorpion Mesobathus tamulus. As you can see, the primary sequence has the inherent active site motif that translates to the three-dimensional arrangement of residues that helps in catalysis, which in turn dictates the function at a particular pH. Therefore, it is quite evident that the participation of histidine, which is seen in the primary sequence and at the three-dimensional structure level, is inherently active at the optimum pH of 8. Therefore, we see a two-way directional relationship between the sequence of the protein, the three-dimensional structure, and the functional outcome of a particular protein. Now, coming to the second example. For the calcium dependency, you see that you have what you call as the calcium binding loop or a motif that is well represented and, more importantly, well conserved across a series of sequences of the same family from different organisms. This conserved loop is also maintained at a three-dimensional structural level, where you see a very beautiful arrangement of glycine and tryptophan alongside the aspartate 34, which contributes carboxyl oxygens, thereby coordinating the binding to the calcium ion. What is also important is, alongside the coordinate bonds, this particular loop is stabilized by the disulfide bond between the loop to a neighboring helix. This in turn translates to a function where the enzyme is dependent on calcium, or in other words, calcium becomes a cofactor for the catalytic activity of this particular phospholipase A2 protein. Thereby, the function is a resultant of the three-dimensional structure, which in turn is a resultant of the sequence. This very beautifully illustrates that sequence structure function relationship is very integral, well maintained, and useful for understanding proteins. Protein structure prediction also becomes important from the perspective of characterizing a protein as a drug target. For example, the human group 3 phospholipase A2 is a potential drug target for inflammation, atherosclerosis, and cancer. Therefore, an understanding of the three-dimensional entity becomes very integral to designing small molecule inhibitors. And when you talk about the structural characterization, you could be looking at understanding important functional motifs such as the calcium binding site, the active site where the catalysis takes place, and finally the substrate binding channel, or the channel in which the substrate binds to the enzyme. Protein structure prediction becomes important for drug designing. In this example, I have shown that a understanding of the drug target from a three-dimensional perspective becomes very important to understand the intermolecular interactions that could possibly be between the ligand and the protein, the enzyme substrate interactions, and finally the protein drug interactions. The protein drug interactions comprise of all the non-covalent interactions that could possibly exist between the molecules and the amino acids. More the number of interactions, more the potency of that particular drug molecule against this particular target of group 3 phospholipase A2. Structural basis of a disease is another important application for protein structure prediction. We here see the structures of glycine 80 variant and the serine 80 variant of the human group 2D phospholipase A2 along with its receptor complex. That paves the way for understanding the weight loss in chronic obstructive pulmonary disease. Without going into details, what is important is the fact that the serine 80 variant has a stronger interaction with the receptor as seen by the interacting surface shown in the red color as compared to the interacting surface on the enzyme molecule of the glycine 80 variant, which is slightly lesser. What does that mean? It means that serine 80 is a more potent ligand for this particular receptor, thereby triggering the release of cytokines that cause or mediate weight loss in patients with chronic obstructive pulmonary disease. Understanding drug toxicity also is an important application that could be used by protein structure prediction. Gentamicin, a 3-carbohydrate ring moiety, binds to secondary targets such as calreticulin, thereby competitively inhibiting the binding of unfolded proteins to this particular chaperonic protein, thereby causing an accumulation of unfolded proteins and resulting in nephrotoxicity. This is very well illustrated here, and the details of which is available in the published paper mentioned below. Protein structure prediction also helps to understand the evolutionary background of a particular protein. Here, in our lab, we tried understanding the sequences of group 3 phospholipase A2 from an array of organisms from insects to giga monster, rabbit, the human body, the bat, the sea anemones, fishes, and the scorpion. What is interesting is the catalytic site is different in different organisms. It therefore gives a perspective, and it also gives an important understanding to the functional use of this particular enzyme in that particular organism. It may be a component of the venom, it may be a component of the salivary gland to digest food, or it could act as a housekeeping function in various organ systems in the body. Protein structure prediction as a tool to develop personalized medicines. Different people respond in different ways to the same medicine. Why is it so? That is because people have certain enzymes whose three-dimensional structure may slightly vary from one another. Therefore, they could be having different isoforms for the same type or group of enzyme. When the sequence of the enzyme or there is a slight variation to the structure, it implies that you need to design different potent molecules that could ably bind to different isoforms in the respective individuals. In this example, A, B, C, D represent the catalytic site or the site where the substrate binds on this particular enzymatic drug target. And very rightly, we have shown that different molecules bind with different potencies to each of these 4 different isoforms. This lies the concept or the foundation for the concept of personalized medicine. So, students, hope you enjoyed this lecture. Let us now summarize what we have already learned in this particular module. Protein folding has a lot of relevance on the structure and its aspect on function. Protein structures are experimentally determined using various techniques, but all these techniques have their own limitations. DNA and protein sequences are a large data pool, and in addition to experimentally solved structures, will help one to solve more unsolved protein structures. Though there is the use of the word prediction, the entire principles of computational based prediction is based on established biophysical principles and mathematical calculations. And protein structure prediction has an array of uses and applications in biology, health, and medicine. Thank you.